I want to tell you about a story that stayed with me long after I first read it. A 58-year-old woman, a retired schoolteacher. Meticulous about her health, she walked every morning, avoided fast food, cooked almost everything from scratch. And yet, despite doing so many things right, she was diagnosed with stage 3 pancreatic cancer. When her physician traced back through her medical history and dietary habits, he found something striking, a set of completely ordinary kitchen behaviors. Things nobody had ever warned her about. That is why I created this video. My name is Adriana Markson. I am a health and nutrition researcher with a background in biochemistry and metabolic health. Over the past several years, I have spent hundreds of hours reviewing peer-reviewed literature on pancreatic function, gut health, and the dietary factors that emerging research suggests may contribute to cellular stress in the digestive system. Published in journals including Gut, Gastroenterology, Food Chemistry, and Cancer Epidemiology, Biomarkers and Prevention. What I have found is a pattern that is not getting nearly enough attention outside of academic circles. And today, I want to bring that research to your kitchen table. I am going to walk you through 5 specific foods that undergo potentially significant chemical changes the second time they are exposed to heat, and explain what current research suggests those changes may do to one of the most vulnerable organs in your body, your pancreas. Stay with me through all 5. The 5th involves 2 simultaneous processes that, based on the available evidence, I believe deserve serious attention. Most people watching this are almost certainly reheating it multiple times a week. Let us begin. Before we look at the foods, it helps to understand why the pancreas is at the center of this conversation, because once you grasp the organ's biology, everything else will make immediate sense. Your pancreas performs 2 completely separate jobs. The first is endocrine. It produces insulin and glucagon, the hormones that regulate blood glucose and fat storage. The second is exocrine. It secretes digestive enzymes, amylase, lipase, protease that break down every meal you eat. These 2 functions happen in parallel every hour of every day. Here is what makes the pancreas particularly vulnerable. Unlike your liver, which has extraordinary regenerative capacity, the pancreas has very little. Pancreatic cells, particularly the insulin-producing beta cells and the enzyme-producing acinar cells, do not regenerate efficiently after damage. When they are lost, they are largely gone. The pancreas also sits behind the stomach in the same circulatory territory as the gut. Everything absorbed through your intestinal wall passes through the portal circulation, which runs directly through pancreatic territory. Every compound your diet generates reaches the pancreas with remarkable efficiency. And critically, the pancreas contains no pain receptors capable of warning you when cellular stress is accumulating. There is no alarm system. It works in complete silence, which is a key reason why pancreatic disease is so often detected late. The 5-year survival rate for pancreatic cancer remains below 12%. Not because medicine has failed to try, but because by the time symptoms appear, the window for effective treatment has often already closed. This is not meant to alarm you. It is meant to explain, clearly and honestly, why the research I am about to share carries the weight that it does. The first food on this list is not a specific dish, it is a category. Leftover fries. Refrigerated chicken pieces re-crisped in a pan. The spring roll, the samosa, the battered fish, anything cooked in oil the first time and reheated in oil, or in its own residual grease, a second time. The cooking oils that dominate most kitchens, sunflower oil, corn oil, soybean oil, generic vegetable oil, are polyunsaturated fats with a high concentration of omega-6 linoleic acid. Their molecular structure contains multiple double bonds, making them chemically unstable under thermal stress. When these oils are heated the first time, oxidative degradation begins. This process generates a class of compounds called reactive aldehydes, most notably 4-hydroxynonenal, or 4-HNE, and malondialdehyde, known as MDA. A 2015 study in food chemistry, led by Professor Martin Griffeld at De Montfort University, found that reheating vegetable oils even once may generate aldehyde concentrations that significantly exceed World Health Organization's safe daily thresholds. When you refrigerate that fried food and apply heat a second time, the second heating cycle can accelerate aldehyde generation. Some research suggests concentrations 2 to 4 times higher than during the initial cook. Once absorbed through the intestinal wall, 4-HNE and MDA may trigger inflammatory cascades, generate reactive oxygen species, and, according to research published in the journal Carcinogenesis, potentially form DNA adducts with guanine bases, the type of modification associated with mutations in the p53 tumor suppressor gene, which is frequently found in pancreatic adenocarcinoma. I want to be precise here. Most of this evidence comes from laboratory and animal studies. Direct causal links in humans require further research. But given the biological plausibility and the simplicity of the fix, the science is worth understanding. The solution is straightforward. Reheat fried food dry in an oven at 180 degrees Celsius or in an air fryer so no fresh oil is oxidizing under heat. The texture returns. The chemical concern does not. And consider treating your frying oil as single use. The cost of a bottle of oil is not comparable to what we are discussing. The second food is the one that genuinely surprises people. Spinach, kale, Swiss chard, arugula, beet greens. These appear on every anti-cancer diet list ever published for good reason. They are genuinely protective foods when consumed correctly. The key word is correctly. When these foods are cooked and then reheated, specifically after hours of refrigerated storage, a chemical transformation may occur that is worth understanding. All leafy green vegetables contain inorganic nitrates absorbed from soil. In their fresh or freshly cooked state, these nitrates are generally harmless. The human body actually uses small amounts of dietary nitrate to produce nitric oxide, which supports vascular health. But when cooked greens sit, even under refrigeration, naturally occurring bacteria begin enzymatic conversion of nitrate to nitrite. Research published in the European Journal of Nutrition has documented meaningful nitrite accumulation in cooked leafy greens within just 4 to 6 hours of refrigerated storage. Now apply heat a second time. Those accumulated nitrites may react with amines present in the food to produce a family of compounds called N-nitrosamines. The International Agency for Research on Cancer classifies nitrosamines as probable human carcinogens, Group 2A. Epidemiological data published in the American Journal of Epidemiology have identified associations between high nitrosamine exposure and elevated pancreatic cancer incidence, though these findings are observational, and causation has not been definitively established. The important clarification: cooked leafy greens eaten immediately are not a significant source of nitrosamines. The concern arises specifically from the storage then reheat cycle. This is a preparation problem, not an ingredient problem. The fix is simple. Eat cooked greens immediately. If they must be stored, eat them cold the next day. Cold cooked spinach in a grain bowl retains every nutrient and generates no nitrosamines because there is no second heat event. Each additional heat cycle compounds the concern multiplicatively. The third food is rice. And of all 5, this is the one that causes the most resistance when I explain it. Because rice seems so safe, so neutral, so inoffensive. But the danger in reheated rice has nothing to do with the rice itself. It has everything to do with a remarkably resilient microorganism called Bacillus cereus. Bacillus cereus is naturally present in raw rice from virtually every supply chain. This is not a contamination problem, it is simply a characteristic of the grain. When you cook rice, boiling water kills all active bacteria. But Bacillus cereus produces endospores, a dormant biological form that survives temperatures of 100 degrees Celsius for extended periods. Standard cooking does not eliminate the spore load. When cooked rice begins to cool, those spores respond to the falling temperature exactly as they evolved to do. Between approximately 10 and 50 degrees Celsius, which encompasses your kitchen counter as the rice sits after cooking, the spores germinate, the bacteria emerge from dormancy, and they begin producing toxins. The toxin central to our concern is cerulide, heat stable up to 121 degrees Celsius. Your microwave does not reach that temperature. Your stovetop reheating does not eliminate it. Standard reheating kills the bacteria, it destroys the factory. But the product the factory already produced remains fully intact in every bite. Most people experience mild bacillus serious exposure as nothing more than brief nausea and then recovery. What they do not know is that subclinical exposure, doses below the threshold of obvious illness, may still trigger a measurable inflammatory response in the gut mucosa. And this is where the gut-pancreas axis becomes critical. Research over the past decade has substantially advanced our understanding of the bidirectional signaling network connecting intestinal health to pancreatic function. When gut epithelial cells respond to microbial toxins, pro-inflammatory cytokines, including TNF-alpha, interleukin-6, and interleukin-1 beta, are released into the shared circulation. The pancreas receives those signals. And when this cycle repeats regularly, the cumulative inflammatory burden on pancreatic tissue accumulates in ways that a 2019 review in the journal Pancreatology linked to elevated pancreatic cancer risk. The fix: cool cooked rice within 1 hour of cooking by spreading it thin in a wide, shallow container. Refrigerate immediately. Consume within 24 hours. Reheat to steaming hot throughout, not warm, not hot to the touch on the outside, but visibly producing steam from the interior. And do not return it to the refrigerator after reheating. The fourth food is chicken, the most consumed poultry on the planet. And the way it is most commonly reheated, in a microwave, tested with a finger touch on the outside, declared done, is one of the most reliably deceptive food safety practices in common use. A microwave oven heats food through dielectric heating. It excites polar water molecules, causing them to vibrate and generate heat through friction. This mechanism is fundamentally dependent on the water content and density of whatever it is acting on. A piece of chicken thigh is not a uniform material. It has fat deposits, muscle tissue, cartilaginous areas, and bone, each with a different water content. The result is uneven heating. Not mildly uneven, dramatically uneven. Research published in the Journal of Food Protection has documented internal temperature differentials of 20 to 30 degrees Celsius across different regions of the same microwave reheated chicken piece. The outside edge of a thigh can reach 75 degrees, while a region 2 centimeters inward, adjacent to bone, sits at 50 degrees or below. Those interior pockets are not death zones for bacteria. They are incubators. The two pathogens that concern me most in this context are Salmonella enterica and Campylobacter jejuni, both naturally present in raw poultry, both eliminated by thorough initial cooking, and both capable of recontaminating cooked meat through post-cooking handling. Campylobacter jejuni in particular has been studied in relation to persistent gut mucosal inflammation. Research from the Karolinska Institute has examined associations between repeated Campylobacter exposure and chronic gut inflammatory states, the kind that researchers have been linking to elevated gastrointestinal cancer risk, including pancreatic cancer, through the inflammatory cytokine mechanisms I described earlier. Even without a dramatic illness event, repeated subclinical exposure may maintain the gut in a state of immune activation, allow bacterial products to translocate across a compromised gut wall, and reach the systemic circulation where the pancreas, sitting in that same circulatory territory, may respond with its own inflammatory signaling. The fix: reheat chicken in an oven at 175 degrees Celsius, covered tightly with foil, for 15 to 20 minutes. The enclosed, moist heat environment eliminates cold spots by heating the entire piece evenly from all directions simultaneously. If a microwave must be used, cut the chicken into small, roughly uniform pieces, use medium power, stop halfway through to rearrange, then leave it covered for a full 2 minutes after the microwave stops. The carryover heat during that rest period is what finishes the job. Or simply eat it cold. The fifth food is reheated potatoes. I have saved this one for last because it is, in my assessment, the most biochemically complex situation on this list, and because most people who hear the first 4 foods think they are safe with potatoes. Based on the current research, that assumption deserves a much closer look. Reheated potatoes, specifically potatoes that were roasted, fried, or baked the first time and are being subjected to high heat again, may carry not one chemical concern, but 2, occurring simultaneously through completely independent mechanisms. The first is acrylamide accumulation. Acrylamide is a chemical that forms when starchy, carbohydrate-rich foods are exposed to temperatures above approximately 120 degrees Celsius. The mechanism is a Maillard reaction, a condensation reaction between the amino acid asparagine, present in high concentrations in potatoes, and reducing sugars. The golden color and roasted flavor that make a properly cooked potato so enjoyable are, chemically, the visible signature of this same process. The International Agency for Research on Cancer classifies acrylamide as a probable human carcinogen, Group 2A. In pancreatic tissues specifically, acrylamide's primary metabolite, glicidamide, has been shown in laboratory research to form DNA adducts with N7-guanine and N3-adenine, classes of mutagenic lesions associated with acinar cell transformation, one of the cellular precursors to pancreatic cancer. Here's what makes the reheating scenario specifically more concerning than the original cook. When a roasted potato is refrigerated overnight, moisture evaporates and sugars and asparagine concentrate in the dried outer layer. When high heat is applied a second time, the substrate conditions for acrylamide formation, high asparagine concentration, high reducing sugar concentration, and low water content are more favorable than they were during the first cook. Research published in Food Chemistry has documented that refrying previously cooked potatoes may produce acrylamide concentrations up to 50 to 60% higher than those generated during the initial frying event. The second concern operates through a completely independent mechanism, and it concerns the slow, cumulative stress on pancreatic function through glycemic load. When a cooked potato cools fully to refrigerator temperature, a process called starch retrogradation occurs. The amylose chains in the cooked starch realign into a tightly packed crystalline structure, resistant starch type 3. This is a genuinely beneficial transformation. Resistant starch resists digestion in the small intestine, meaning it does not trigger rapid glucose absorption. It passes into the large intestine, where it acts as a prebiotic fiber. Its glycemic impact is dramatically lower than that of freshly cooked starch. A cold-boiled potato has a glycemic index approximately 25 to 30% lower than the same potato eaten hot. Now apply high heat. In approximately 90 seconds of microwave exposure, the crystalline retrograde structure is disrupted. The hydrogen bonds collapse. The starch reverts to a rapidly digestible form, high glycemic, high insulin stimulating. Pancreatic beta cells respond to that glucose surge with an acute insulin secretion event. Once, this is completely manageable, but repeated chronically, 3 times a week, every week, over years, the pattern matters. Chronic overactivation of insulin secretion has been associated with progressive beta cell stress, declining insulin secretory capacity, and a metabolic trajectory linked to elevated pancreatic cancer risk. A 2017 analysis published in Cancer Epidemiology, Biomarkers and Prevention identified elevated fasting insulin as an independent risk factor for pancreatic cancer after adjusting for BMI, diabetes status, and other confounders. And if you are refrying previously cooked potatoes, adding the acrylamide accumulation of the first concern to the glycemic spike of the second, and adding on top of that the oxidized oil aldehyde exposure from food number 1, you are stacking multiple concerns from this list into a single dish. The fix: eat cooked potatoes cold whenever possible. Cold potato salad with extra virgin olive oil and fresh herbs is not a sacrifice, it is a fundamentally different metabolic event. You preserve the resistant starch, generate no additional acrylamide, and give your pancreatic beta cells a meal that asks almost nothing of them. If warmth is needed, steam gently or use the minimum heat that takes the chill off. Never refry a previously cooked potato. I want to be careful about how I close this, because information like this, delivered without context, can feel paralyzing. That is not the intention. That has never been the intention. What I have described today is not a sentence. It is a pattern, and patterns can change. Chronic inflammation can be reduced. Oxidative stress can be modulated through antioxidant-rich dietary choices, particularly polyphenol-rich foods like extra virgin olive oil and berries, which activate the NRF2 pathway, your body's master switch for antioxidant enzyme production. The gut microbiome can be supported through probiotic and prebiotic foods. DNA repair capacity can be bolstered through adequate folate, zinc, and selenium. Every better choice you make from today forward represents a measurable shift in the right biological direction. Here is the condensed protocol I want you to carry out of this conversation. Fried foods, reheat dry, oven at 180 degrees, or in an air fryer. No fresh oil. Treat frying oil as single use. Leafy greens. Cook only what you will eat immediately. Store leftovers cold and eat them cold. Never apply a second heat cycle. Rice. Cool within 1 hour in a wide, thin layer. Refrigerate promptly. Eat within 24 hours. Reheat to full steaming temperature. Never refrigerate after reheating. Chicken. Oven at 175 degrees with foil. If using a microwave, cut into uniform pieces, use medium power, and rest covered for 2 minutes after heating. Or eat it cold. Potatoes. Eat cold whenever possible. Never refry a previously cooked potato. If warmth is needed, use gentle, low heat. The research exists. The gap between that research and the kitchen table is one that each of us, one conversation at a time, has the power to close. Take care of yourselves. I will see you in the next one.